In this video, I'm going to talk about non-Mendelian genetics, or some exceptions to the rules about what we've been talking about recently with our monohybrid problems. As you watch this video, you should have your notes split screened on your iPad, so you're filling out anything that's filled out on mine that's not filled out on yours. So here are some exceptions to the rules. A lot of traits have more than one gene. What we've been doing with monohybrid problems is looking at one gene and crossing it. There's some that have more than one gene that's controlling that trait. Some genes have two or more alleles to represent one trait, and not all genes have only a dominant or recessive form. So we're going to talk about all of those in this video. Traits that are controlled by more than one gene are called polygenic trait, and think of poly for many. We're going to have a range of traits when it's a polygenic trait. Like skin color is decided by 5 different genes that are coding for a pigment or a protein that codes for your melanin that gives you your skin color. So many genes give us a range of different traits like skin color and eye color, hair color. Another example of polygenic traits is height. We don't have just your 5 foot, 5'5", or 6 foot. There's a range of heights all in between there. So this is considered a polygenic trait because it is more than one gene controlling how tall you are. Some genes have multiple alleles, 2 or more alleles, that are going to code for a single gene. So you're going to end up with 3 or more phenotypes. So you see in this example with the bunnies, they could have full color, they can be chinchilla color, they can be Himalayan color, or albino. And you see that there are 4 alleles that can code for these colors. So different from what we've been doing where we have big B for black and little b for white, there are 4 letters that these could be. So I mentioned that range of color for hair is polygenic, but it's also multiple alleles. Brown hair shows multiple alleles, and we also have this range of all of these colors that it could be. We'll go more into depth about blood typing tomorrow, but blood typing is an example of multiple alleles too. You can have an allele for A, B, AB, and another type of blood that you can have is O. It's also codominant, and we'll talk about that later on about what that means. But this means you can have 3 different alleles, and we can have 4 different blood types. Not all genes have a dominant or recessive form. So we're going to talk here about incomplete and codominance. And these are the types of Punnett square problems that you're going to complete for your homework after this video. So you have incomplete dominance. You see if this red flower and this white flower have offspring. Incomplete dominance is a mix of these 2 colors to give us pink. If we talk about codominance, we have this red flower crossed with a white flower, and we see both traits at the same exact time of red and white. That is codominance. When we define incomplete dominance, we're going to say that incomplete dominance, the heterozygous gives us a mix. So like in this example, we have red flowers, we have white flowers. If you mix red and white together, you get pink. So for the genotypes, red flower, we're going to give big R, big R. White flower would be little r, but the heterozygous is pink, that is big R, little r. This problem here is a generational problem. Where we start out with a homozygous red and a homozygous white to get the F1 generation to get the F2 generation. So if my genotype for red is big R, and my genotype for white is little r, we could put this into a Punnett square, but we see that this pink flower is going to get a big R from one parent and a little r from another parent. Every single one of them is heterozygous in the F1 generation. We cross an F1 with an F1, and we can get all different combinations. In a Punnett square, we can look here. Big R is going to be your red. Big R, little r, the heterozygous, is now pink, the mixture between red and white. And little r gives us our white. So we see 1 out of the 4 being red, 2 out of the 4 being pink, and 1 out of the 4 being white in this F2 generation. For codominance, the heterozygous is when you see both traits at the same time in like spots or speckles or stripes. So an example would be spots. So if I say black has a genotype of big B, white is little b, the heterozygous would be big B, and we would see both black and white. So codominance, think of it this way. Co means like they're cooperating together. If you are ever in a classroom that has a co-teacher, so you see 2 teachers at the same time, that's called co-teaching. Codominance, they're seeing both of them at the exact same time, usually in spots, speckles, or stripes. For this problem, we have these cute little poofs, and they come in color combinations. And I want to see if these 2 poofs showed incomplete dominance, what would their baby look like if it was heterozygous? And also taking these 2 poofs and showing codominance if their baby was heterozygous. So you're going to draw for both of these. Stop and pause your video, do your little drawing, come back to this video to see what the right answer was. If you're showing incomplete dominance, blue is not completely dominant over yellow, yellow is not completely dominant over blue, so a mixture of blue and yellow is a green baby poof. For your codominance, again, blue is not dominant over yellow, yellow is not dominant over blue, but they're going to work together, they're going to cooperate together. So you see this in spots, blue and yellow spots. Let's put this into a Punnett square. So it says gold is crossed with a silver unicorn and all of them are bronze. Because we're getting a mixture of them, gold is not completely dominant over silver, silver is not completely dominant over bronze. We call this incomplete dominance. When you define your alleles, what are the letters going to represent for each of these colors instead of what we've been doing with 2 options? This time we need to give all 3 options. So I'm going to say, you can use any letters you want. I'm going to say that big G is gold. I'm going to say that little g is silver. But the heterozygous big G, little g is going to be bronze. My incomplete dominance, my mixture. So it says gold crossed with a silver. So big G is crossed with little g. And I put this into my Punnett square. Big G, little g, one parent along the top, one parent along the side. And these boxes show fertilization, all of the options. The genotypic ratio, same thing as what we've been doing before, 0, big G. I see all 4 of them as big G, and 0 of them as little g. But when we get to the phenotypic ratio, there's a little bit of a difference here. We need to list out all 3 options. So I see 0 gold. I see 4 bronze, 4 out of the 4, 100% of the time they're bronze, and 0 silver. So notice I have 3 traits here listed in my phenotypic ratio. Let's try another example. Gold unicorn is crossed with a silver unicorn. Now all the offspring have silver and gold spots. Because I see both traits at the exact same time, we call this codominant. They're cooperating together. So if we cross 2 spotted unicorns together, we want to know the genotypic and phenotypic results. Let's define those alleles. I'm still going to use big G for gold. little g for silver, and the heterozygous for spotted. So if I'm crossing two spotted unicorns, their genotypes are big G crossed with big G little g. I put one parent along the top and one parent along the side, and each of the boxes show all of the options, possibilities in fertilization for their offspring. Genotypic ratio, I see 1 big G. I see 2 big G, the heterozygous, and 1 little g. My phenotypic ratio, I need to list out all 3 possibilities. So I see 1 out of the 4 could be gold, 2 out of the 4 could be spotted, and 1 out of the 4 silver. Now that you've watched this video, you're going to jump into Class Kick. And in Class Kick, you're going to do the assignment for the incomplete and codominance problems. This video and these notes can help you with that if you ever get stuck. So you're going to go ahead and complete that homework assignment. If you have any questions about any of this stuff, bring it to class tomorrow or email me with any questions that you have. Thank you for watching.